Good afternoon, students. Myself, Dr. Ghanshyam Chavan. Today, our part of discussion is cell junctions. What are these cell junctions? Students, cell junctions are nothing but the point of attachment between two cells. So, mostly the epithelial cells are connected with each other, the muscular cells are connected with each other. So, by the cell junction, they are connecting one cell with the another cell. And if you will see, there are many tight junctions between these cells, and these cell junctions are contact between point of plasma membrane and tissue cells. Now we have to know the terminologies of cell junctions which are used. In that, first is catherines. These catherines are nothing but the molecules. These are adhesive molecules which are forming the adherent junction, and they are responsible for the adhesion of one cell with the another cell. So these molecules are attachment of cell from the another cell, and these catherines are always connecting the cell together. Next terminology is integrins. Integrins are nothing but the transmembrane receptor proteins, and again these receptor proteins are responsible for connection of one cell with the another cell, and it is also responsible for connection of cell with the extracellular matrix, and they are forming the bond between that cell and the extracellular matrix. Tight junctions. So tight junctions are nothing but the occluded junctions. They are also called as narrow junctions. They are very close junctions. They are producing junctions between the two plasma membranes, and these tight junctions are produced by uh, pro, uh, the protein complex junctions, and these junctions are responsible for production of the one cell to the another cell attachment by the help of the various transmembrane proteins. And the specialty of this tight junction is that to prevent the leakage of the solutes which occurs between two cells, and it also seals the paracellular transportation or paracellular contact. Now we will switch over to the desmosomes. The desmosomes are nothing but the specialized proteins, and these proteins are responsible for attachment of or adherent of one cell with the another cell. And these desmosomes has been discovered by the scientist Joseph in 1920. And in these desmosomes, the actin are attached with this cadherin. They are forming the bond between these two molecules. Adhesion belt. The adhesion belt is nothing but it is an attachment between two cells occurs by the molecules, and they are forming the vertebra like structures. And these adhesion belt are nothing but the junctions. They are the additional junctions or supplementary junctions, and they are forming the band like structures. The typical band like structures they are forming when they are connecting between the two cells. And the hemidesmosomes, these are just look like the desmosome if you will see by under the microscope, but they are nothing but the stud like structures, and they are specialized cells. They are proteinous cells. These are available at the keratins of the skin. The skin epidermal cell contains these hemodesmosomes, and their specialty is they are connecting with the extracellular matrix. It is a basic difference between hemidesmosomes and the desmosomes. And if you will see desmosomes, they are connecting between one cell to another cell, means they are connecting two adjacent cells whereas these hemidesmosomes they are connecting with the extracellular matrix. Next is gap junctions. What is gap junctions? It is nothing but the protein complex junctions, and these gap junctions are required for the connecting to cytoplasm of two different cells, and it is very free junctions, and it is responsible for transmission or transportation of the various molecules of the various solutes. Also they are transferring the various electrolytes, they are also transferring the various impulses from one cell to the another cell very freely. And the last is connexins. What are these connexins? Connexins also, they are nothing but the protein of gap junctions. They are nothing but the adhesive material or adhesive protein of these gap junctions. So they are responsible to connect one cell with the another cell, and they are forming the vertebra like structure. They are producing the bond, or they are producing the attachment between two cells. So number of attachment will be produced. They are nothing but the transmembrane proteins. So they are connecting two cells very closely, and they are producing the vertebra like structure. This number of the transmembrane proteins will be attached to the two cells, one side by the another side. Now overview. Multicellular organisms combine cell into tissues. Extracellular matrix is a complex network of molecules, proteins, glycosaminoglycans, proteoglycans secreted by cells. Specialized extracellular matrix include connective tissue, basal lamina, exoskeleton, cartilage or bones. Tissue are formed from cell-cell connections and cell matrix connections. Now here in this diagram, students, you can see it is a diagram of histology of gut of small intestine, where you can see the villi, macrovilli, which are prepared by the epithelial cell. So that when one cell is connected with the another cell, it is also done by these junctions, very junctions that we have come across recently, and also we will discuss later on in details. There is epithelium cell, connective tissue, they are also connected with each other with the various junctions, circular fibers, longitudinal fibers, connective tissue and epithelium. So in this fashion the various layers are available there, and in abruptly if you will see, there are having four basic layers, for example mucosa, submucosa, after that adventitia and serosa. So in this fashion there are four layers that we should know. Now we will switch over to the cell junctions. Now one by one in details we will see that cell junctions. So in that first is tight junctions, adherent junctions, desmosomes, hemidesmosomes and gap junctions. Now occluding junctions, it is also called as tight junctions. So it is tight junctions of intestinal epithelium. So in this figure you can see another figure of lumen of gut. Lumen means internal part of our gut. Gut means intestine. It will be small intestine, large intestine. So internal round hollow part of your intestine is called as lumen of gut. So in that, if we will do the section of that lumen, so we will come to know this structure. This is the structure. So where the transportation means whatever food we are taking inside our body, we are ingesting, we are eating. So that food material, from that the nutritional or electrolytes or the nutritional value or glucose or carbohydrate, that will get incorporated into our blood. So their transportation occurs in this fashion. So they are coming from the apical membrane towards our blood, towards our systemic blood circulation. So here how the transportation will occur, that you can see here in this figure, the tight junctions are there, the plasma of membrane of adjacent cells are also there, intercellular space is there, passive glucose carrier proteins, because for the transportation of protein the carriers are required. So GLUT4 like that carried proteins are there which are carrying glucose from the apical membrane towards our systemic blood circulation. So their glucose is carried out from one point, means at the edge of your apical membrane towards your blood vessels. So in this fashion, the continuously the transportation of is occurs from that apical membrane towards your extracellular fluid. That's why at the middle edge, at the middle part of this figure, if I see, the high glucose concentration is there. Tight junctions. Each cell possesses integral membrane points that bind to the similar proteins in the adjacent, forming a continuous weld. These tight junctions consist of wave-like strands of transmembrane proteins that fuse together the outer surface of adjacent plasma membrane to seal off passageway between adjacent cell. So these two adjacent cells, if you will see, they are very close to each other, and they are joined by these transmembrane proteins which are doing sealing of these paracellular cells, and they are producing the tight junctions. So they are not allowing the leakage of any solute which are transporting from in the parallel way. So in this fashion, the cells of epithelial tissue that line in the stomach, intestine and urinary bladder have many tight junctions. So because these intestine, urinary bladder or stomach, we cannot waste any material from that. We are not compromise for wasting of any solute or any nutrients or any electrolytes. That's why the tight junctions are very required. That's why whatever the HCl has been secreted into our stomach, but it won't transport it, or it won't go away, or it will bisect our mucosal level. It will be stay there. So that's why it won't allow to any leakage. That's why these tight junctions are working very properly in our body. They inhibit the passage of substances between cell and prevent the content of these organs from leaking into the blood or surrounding tissues. Here you can see the calodin or occludin is there. They are responsible for these tight junctions. The transparent proteins are there. They are forming these tight junctions. Anchoring junctions. It is a part of these tight junctions. Integral membrane proteins connects a cell's cytoskeleton to another cell or extracellular matrix. So in this figure you can see how these cytoskeletal filaments are connecting, and they are producing these anchoring junctions. Integral membrane proteins connect a cell's cytoskeleton to another cell or extracellular matrix. So here the intracellular anchor proteins that you can see, so they are responsible to connect with these transmembrane adhesion proteins. So these transmembrane proteins are doing the connection between these intracellular anchor proteins, and they are responsible for transportation of any information or solute from one cell to the another cell. Anchoring junctions. The cytoskeletal fibers connect to a membrane protein receptor which attaches to another protein in either the extracellular matrix or another cell membrane. Now catherins or desmosomes, each attach cell to cell, and they are specialized proteins which are attached in between two cells. They are forming band between two cells, and they are made by these catherins. They are the specialized molecules, so they are joining two cells. These receptors extend out from the cell, binding to other catherins. If you see this desmosome, recently I have told that it has been discovered by the scientist Joseph in 1920, and they are like the adherent junction. Desmosomes contain plaque and have transmembrane glycoproteins that extend into intracellular space between adjacent cell membranes and attach cell to one another. So they are attaching cell with the one another. and they are also having these transmembrane proteins. However, unlike adherent junctions, the plaque of desmosomes does not match to microfilaments. Instead of that, a desmosome plaque attaches to the element of the cytoskeleton known as intermediate filaments, which consist of protein keratin. So they are bind with the elements of cytoskeleton, which is known as intermediate filaments. The intermediate filaments extend from desmosome on one side of the cell across the cytosol to desmosomes on the opposite side of the cell. So this structural arrangement contribute to the stability of the cell. That's why it's giving the stability to the cell, and it will increase the strongness of the bond and tissue. These spot weld-like junctions are common among the cells that make up the epidermis. So mostly in case of the keratinized cells of skin epidermis, there you will find these desmosomes. And among the cardiac cell also, in heart also, in cardiac cell you will find this typical type of these desmosomes, because these protein desmosomes are also required to continuously pumping of the heart, and these typical binding between two cells are required to completely relax and to completely contract your heart. The skin also. Now desmosomes prevents epidermal cells from separating under tension and cardiac muscle cell from pulling apart during contraction. Now Catherine participate in the adherent junctions. So if you see in this figure, you will come to know how these actin filaments inside microvillus are there, and the Catherine are producing these adherent junctions in the lumen of your intestine, where the microvilli extended apical surfaces. Here you can see the Catherines are there. They are joining between the two adjacent cells, and again you will find that tight junction is also there, and adherent junctions are also there. There are bundle of actin filaments. The lateral plasma membrane of the adjacent epithelial cells are also available here. Under the cell membrane, contractile fibers of microfilaments connect to cell membrane proteins called catherins. So they are the specialized molecules which are responsible for connection between two adjacent cells. They surround the cell forming a specialized belt. They are forming the belt-like structure. And the adherent junction, if you will see, they contain the plaque, a dense layer of protein on the side of the plasma membrane that attaches both the membrane proteins and the microfilaments of the cytoskeleton. The transmembrane glycoprotein called cadherins, they are joining this cell. Each cadherin insert into the plaque from the opposite side of the plasma membrane partially crosses the intracellular space. So they are not completely crossing, they are partially crossing that intracellular space between these cells and connect to the catherin of the adjacent cell. In epithelial cells, adherent junction often form extensive zones called adhesion belt because they encircle the cell similar to the way of belt encircle your waist. So whatever the belt we are wearing around our waist, so in that fashion it will encircle or it will wrap that cell around that cell. Adherent junctions help epithelial surface resist separation during various contractile activities and when the blood moves through the intestine. So it won't allow the separation of that cell from one another. Now desmosomes, catherins also form localized spot connections. So catherin attaches to the intermediate filaments via anchoring proteins, a desmosomes. So recently we have discussed regarding the desmosome. So again we will discuss that. So how these keratin filaments are there, are made of proteins. So how they are producing or anchoring toward the cytoplasmic plaque formation, and they are interacting between the two plasma membranes. By these specialized proteins, they are joined together two cells, and there are also cytoplasmic plug made of intracellular anchor proteins. Here you can see the desmoglein and the desmocholine. They are the specialized proteins are there. They are attached by these intermediate filaments with the help of this placoglobin. It is also another protein, and the desmoplakin, it is also another specialized protein, and in this fashion they are connecting between these two cells. So they are just like junctions of desmosomes which contains the plaque and have transmembrane glycoprotein that extend into the intracellular space between adjacent cell membrane and attaches cell to one another. However, unlike adherent junctions, the plaque desmosomes does not attach to the microfilaments, and instead a desmosome plaque attaches to the elements of cytoskeleton known as intermediate filaments, which consist of protein keratin. So protein keratin is there, that specialized keratin protein is available into our epidermal cell. The intermediate filament extend from desmosomes on one side of the cell across the cytosol to desmosome on the opposite side of the cell. This structural arrangement contribute to the stability of the cell and tissue. So it provides the strongness, it gives the strongness towards your cell. That's why our skin is too much strong. So whatever rupture or whatever heat or whatever the cold sensation is there, but still it becomes very strong. It won't spoil very easily. These spot weld like junction are common among the cell and that make up the epidermis. It is the most outer layer of the skin, which is very tough. And also among the cardiac muscle of the heart. The cardiac muscle is also very tough, and it hold the whole the blood which comes into that heart, and it contract completely whenever blood have to reach out towards the aorta. So that's why for complete expansion and to complete constriction of the heart, we require that muscles to be very strong, and to strengthen that muscle the desmosomes are having great role. Desmosome prevent the epidermal cell from separating under tension and cardiac muscle cell from pulling apart during contraction. Now after this we will go for the next. It is called cell to cell attachments, focal adhesion and hemidesmosomes. Now hemidesmosomes we will discuss. In this, the cytoskeleton fibers attached to the transmembrane proteins or receptors, they are also called as integrins, that are attached to the extracellular matrix components. It is a specialized difference between hemidesmosomes and the desmosomes, because in desmosomes they are attached to the adjacent near cell, and where in case of hemidesmosomes they are attached to the extracellular matrix components. And focal adhesion use and hemidesmosomes use. The focal adhesion used in MF and the hemidesmosomes are used in the IF. Now in these hemidesmosomes, they are resembles to the desmosomes, but they do not link adjacent cells. The name arises from the fact that they look like half of the desmosomes. However, the transmembrane glycoproteins in hemidesmosomes are integrins rather than catherins. Integrins are nothing but the signaling receptors proteins. So they are transferring the information, they are transferring or they are attaching the two cells by going through the transmembrane proteins. So on the inside of the plasma membrane, integrin attaches to intermediate filaments made up of protein keratin. On the outside of the plasma membrane, the integrin attaches to the protein laminin, which is present in the basement membrane. Thus, hemidesmosomes anchor cell not to each other but to the basement membranes. So here also you will find the keratin cells are also available here, and the laminin protein is also available. There are two types of proteins, keratin and laminin. Try to recall. Now gap junction. It is very important junctions. The gap junctions allow cell to exchange electrical or chemical signals. It is composed of protein that form channels that allow small molecules to pass. Subunit of these channels are connexins that are assembled together to make connexons. The connexons from two cells join together to make a gap junctions. Gap junctions membrane proteins are called connexins. They form tiny fluid filled tunnel called connexons that connects neighboring cells. The plasma membranes of gap junctions are not fused together as in tight junctions, but are separated by a very narrow intracellular space or gap. Through these gap junctions, the solids also are easily transferred because it is a gap junction which formed by the true cells of their cytoplasm. So cytoplasm are easily transferred from one cell to the another cell. And though the connexons, ions and small molecules can diffuse from cytosol of one cell to the another cell, but passages of large molecules such as vital intracellular proteins is prevented. The transfer of nutrients and perhaps waste materials takes place through gap junctions in avascular tissues such as lens, cornea and of the eye. So as we saw, the tears or the waste materials from our body, it can be transferred, and also the firing of neurons means impulse generation and the impulse production and transmission can also pass very easily through these gap junctions. The nutrients, the solutes, the molecules can be easily transported from one cell to the another cell. The gap junctions allows the cell in a tissue to communicate with one another. In a developing embryo, some of the chemical and electrical signals that regulate growth and cell differentiation travel via gap junctions. The gap junctions also enable nerve and muscle impulses to spread rapidly among cells, a process that is crucial for the normal operation of some parts of nervous system and for the contraction of muscle in the heart, gastrointestinal tract and the uterus. So it not only involved into the transmission of impulses from one neuron to the another neuron in case of nervous system, but also it is responsible from the muscle, it either the muscle skeletal muscle or it may be the smooth muscle which occurs in our heart or gastrointestinal tract or uterus or respiratory system. So it allow that transmission of the impulses in the muscles also. So it is having great role to communicate between these cells in the heart or GIT or uterus. Now in this gap junction you can see how it looks, how it works in that figure in very good manner it has been explained. So here the interacting plasma membranes are there, so they are interacting, and this green colored are the connexons in the register forming open channel between the adjacent cell. So they are communicating through the cytoplasm. The channel also you can see, and the connexons is composed of the 6 units. Typical 6 units are available there. So it is just like the receptor, the GABA receptor. So in that you will see the ligand receptor. It is also 5 subunits are there. So here the 6 subunits are there. It is a heterotype. So how it looks, that also you can see actually at gap junctions how these transmembranes are look alike. Now regulations of connectivity. When might a cell want to alter its connections to other cells? How do cells alter these connections? alter the profile of cytoskeletal connections, receptors, mostly receptors are there, and the extracellular matrix. Means whatever the mechanism of our various neurotransmitters are occurs, or contraction relaxations are occurs, or the various secretions of our glands, their secretions or our enzymes which has been occur, it occurs due to these connections, it occurs due to these gap junctions. So, because whatever sensation we are having from the skin towards our brain or from the brain towards your respective organ, so whatever orders has been given or whatever connection has been there, so it occurs only due to these gap junctions. So it alters the binding affinity of receptors. Means you are moving, you are walking, you are thinking, your receptions occurs into our stomach, means secretion of your HCl, secretion of various enzymes, our workout. So all these things, the speed completely depends on the release at other gap junctions, because the receptors are available there in gap junctions. What are receptors? They are nothing but the transmembrane signaling proteins. So they are transfer the information in the form of neurochemicals or the electrical impulses which carries from one neuron to the another neuron. And many are calcium ion dependents. Mostly in case of the muscles, the calcium ions dependence, means calcium is responsible for transferring or contraction and relaxation. So they are transferring the impulses in form of calcium from one muscle to the another muscle. So many are affected by these protein kinase enzymes. In summary, you can see now the tight junctions, how they are looking, how they are having work, we have already discussed. Only in short we will see again tight junctions. They are incorporated between two transmembrane proteins. So between two to the plasma membrane, it is tight junctions have been produced by these transmembrane proteins, and they are not allowing any leaking. Here in case of the adherent junctions, their acting is there. It is nothing but the complex junctional points. Again in case of the desmosomes, how it has been produced by these specialized proteins that you can see, they have been joined together. And gap junctions, how it has been produced, in figure also you can see that it is a junction between cytoplasm of two cells. And the hemidesmosomes, they are nothing but the stud like structures, they are look like desmosomes, but they are attached to the extracellular membrane or extracellular fluid. In this fashion, it is a difference. So, and lastly the basal lamina is there, as you can see in this figure. Now we will summarize the function very shortly in one line, how we can explain their functions, that you can see here. That first is tight junctions. So tight junctions are responsible for seals the neighboring cells together in an epithelial sheet to prevent leakage of molecules between them. And second is adherent junctions. It joins the actin bundle in the one cell to the similar bundle in the neighboring cell. So actin is the element which can be joined with the another cell. They are producing the adjacent cell bond between the two cells. So actin is there. And desmosomes, so joins the intermediate filaments in one cell to those in the neighbor. So from the one cell to the another cell, they are forming the bond by the intermediate filaments like cytoskeleton or another specialized proteins. There is also the role of actin and the cadherin. Gap junctions, which we already discussed, it is the junction or it is the transportation or gap between two cytoplasm of two cells. Between the cytoplasm of two cells, it is freely transportation occurs. Transportations of the electrolytes, nutrients, solutes, as well as the electrical impulses. The impulses which occurs between two cells. For example, in case of the neurons, the impulses has been carried out from one neuron to the another neuron in the synapse. So it also just look like the synapse and allows the passage of small water soluble ions and molecules. And hemidesmosomes, as we saw, hemidesmosomes are different from the desmosomes, and they are connecting the cell and the extracellular matrix and anchors the intermediate filaments in a cell to the basal lamina. So students, in this fashion we have discussed today the cell junctions. So whatever things has been there in these cell junctions, I have tried to clarify all the queries. If any again thing has been remain in your mind, if any doubt is there, you can always call me or you can always communicate with me at any point, at any time. So please be with me and subscribe my channel and share my videos as much as you can, and also like my video. So have a nice day and bye.